So today we are going to discuss heterospory and the evolution of seed habit in Pteridophyta. So first of all, what is heterospory? Heterospory is the formation of different types of spores in megasporangium and microsporangium. Therefore, we are going to talk about the formation of megaspores and microspores. So where did this heterospory came from? It came from the homosporic ancestors of Pteridophyta. In homosporic ancestors, the female gametophyte and male gametophyte, they are formed from the megaspore and microspore within the same gametophyte. It means that there is one gametophyte and both megaspores and microspores, both of them are formed inside the same gametophyte, as we can see in the diagram. Both of them, the egg and the anthrozoid, they'll fertilize to form a zygote, and the zygote will ultimately lead to the formation of sporophytic plant body. Sporophytic plant body is the plant body that comprises of a true root, stem, and leaf. Therefore, we can say that heterospory, which was derived from the homosporic ancestors, in heterosporic condition, what happens is two different types of sporangium, namely microsporangium and megasporangium, both of them are present. So, megasporangium will produce megaspore mother cells, which will undergo meiosis to form megaspores, whereas the microsporangium will produce microspore mother cells, which will produce microspores by the process of meiosis. Both will form the female and male gametophyte. The female gametophyte will produce egg, and the male gametophyte, it will form the anthrozoid. Both will fertilize. Again, same. The fertilization event will lead to the production of zygote and ultimately the production of sporophytic plant body. So, same procedure has been explained here. The heterospory is known to evolve during the Devonian period of the Paleozoic era. These are different living pteridophytes in which heterospory has been observed. Two different types of spores have been observed, such as Azolla, Selaginella, Salvinia, Marsilia, etc. If we look at the plant body of Pteridophyta, like here, we can see the fertile leaves possessing the sporangium. As in the picture here, the fertile leaves possessing the sporangium in groups, that is the sori. Just like this, the picture number 2. In groups, the groups of sporangium are known as sori, and the leaves bearing the sporangium, they are known as sporophyll. The leaves that bear the sporangium in groups, that is the sori, are known as sporophylls in Pteridophyta. The same may be grouped just like this, as we can see in this image. The sporophylls may be grouped, and once they are grouped to form a capsule-like structure, it is known as cone or strobilus. The sporangium, as already discussed, in heterosporous condition would bear two different types of sporangium. As we can see in the image here, microspores, they are produced inside the microsporangium, and megaspores, which represent the female spores, are produced inside the megasporangium. So now we know that homospory is primitive and heterospory is derived from homospory. The heterospory first appeared in plants where each sporangium contained both megasporangium as well as microsporangium. According to Williamson and Scott, 1894, two species of Calamus formed the initial stage that might have led to the heterospory. So there was some incipient heterospory, which later on developed into intermediate heterospory, and then a complete heterosporous condition was observed, for example, Marsilia and Selaginella. In some pteridophytes, for example, Selaginella, a single megaspore that obtained proper nutrition because of the degeneration of other spores in the sporangia has been reported. It formed a pre-ovule during the course of evolution. It developed into a naked ovule in gymnosperms, and then into a covered ovule as we see today in angiosperms. For example, as we can see in the picture here, this female gametophyte is covered inside integuments. Now coming to the evidences that explain the origin of heterospory. Some reports suggest that natural selection is the reason for its origin. Also, the species in which heterospory first evolved, they are now extinct. They don't exist today. The paleobotanical evidences, they suggest that heterospory arose due to the degeneration of few spores and making more nutrition available only to the developing spores. So only a few type of spores developed and survived. According to experimental evidences, they demonstrate the effect of different environmental conditions on the development of spores. For example, low light intensity, that is low intensity of sunlight. If it is low, the sunlight intensity, if it is low, then it will lead to reduction in photosynthesis. Therefore, it leads to production of only microsporangia and not the megasporangia. For example, Selaginella. Same is the case with reduction in temperature. It will lead to the increase in the size of microspores only. The developmental evidences, they make it very clear that most of the spore mother cells in megasporangia, they degenerate and only a single megaspore survives, whereas in the microsporangia most of the microspores, they simply survive and increase in number. Coming to the advantages, or we can say biological significance of heterospory, it is a major step towards seed habit. In heterosporous forms, the endospory gametophytes, they are independent of external conditions. It creates a nutritive environment for the developing embryo, as we see in higher plants. In heterosporous forms, the differentiation of sex can be predicted even at spore stages. So this was all about heterospory.